We're going to continue on with our notes here. Still on Learning Target 2, students will be able to identify the function of the three types of RNA and explain the importance of transcription. So we're going to focus on this PowerPoint on this process of transcription. So you should be able to explain the process of transcription as well as the role of the three types of RNA in transcription. Okay? So we're kind of tying it all together now. So a little visual for you, whenever you're thinking about transcription, you're going from DNA to RNA. You're literally copying the code of DNA onto messenger RNA. And we don't copy everything. We don't go from one end to the other like when we did DNA replication. Then we had to copy all of the DNA so that the new cell got all of the DNA in it. Well, when we're doing transcription, we're only copying the parts that are going to code for proteins because that's what we're going to use that coded message to make as a protein. Well, a gene is a segment of DNA that codes for a protein. So this is the part that gets transcribed. So along your DNA molecule, there are various genes that code for your different traits or for your different proteins. Those are the parts that we need to copy when we're going to go through transcription. And because it involves the DNA, this process still occurs in the nucleus, because remember, DNA cannot leave the nucleus. So here you have a DNA strand. That's going to have to open up so we can copy off the code. But you'll notice, if there's a T on the DNA side, that still codes with an A because RNA still has adenine. But if there's an A on the DNA side, now we have to put a U, uracil, because there is no thymine in RNA. Uracil, the U, replaces the T. Otherwise, C and G still go together, and we just go down the line. Okay? So you still need to know the base pair rules, but there's that little change in that normally where you would have put a T for DNA, now a U gets placed opposite an A. So the first step that has to happen is very similar to what happened in DNA replication. Helicase is going to unwind the DNA by breaking the hydrogen bonds. So we have to separate that DNA. Again, we're not separating the whole strand, just along those segments where the genes are located, the segments that are going to code for a protein. Well, how do we know where those are? There is a region called the promoter. That's the region on DNA that signals the start of a gene that should be copied. It's also going to tell us where RNA polymerase should start copying. I think I covered up my rest of my words with my image here. But you can see here's the promoter region right here, this little area right here. And so we're only going to copy off this segment here where the gene is located. And so helicase would have had to come along, and it would have had to open up this whole area and just kind of open it up so that the RNA polymerase can come and do its job. And I'm going to make note here that the DNA strand, when it's unwound, we only copy the DNA code from the template strand. This is the strand that messenger RNA copies off of. Which sounds weird because the coding strand sounds like that's the one that would have the code, right? Well, when we do some practice, you're going to understand why that is the case. Because if you look here and you see what's copied off, here's your template strand, and the RNA is being copied here, you're going to start to see that these letters are going to look very similar here to what's above. So let's see if I can line it up so you can see this a little better. So like this segment right here coincides with this segment of the coding strand. And see how it's CGC, ACU, CGC, ACT. The only thing that's different, of course, we're replacing the U for the T. But the reason that messenger RNA copies off of the template strand is so that it will look exactly like the coding strand. Remember, messenger RNA has to carry the code now for making a protein to the ribosome. So it better have the same code that the DNA strand had. So we copy off the template strand so that the messenger RNA can look like the coding strand of DNA. So the next step then is for the RNA polymerase, which is an enzyme, moves down the DNA template strand and assembles RNA nucleotides complementary to the DNA nucleotides. Again, U replaces the T. So RNA polymerase has a very similar job as what DNA polymerase did during DNA replication. RNA polymerase is going to move down the line and put the correct nucleotides according to the correct base pairing. And it's always going to use the template strand to copy off of. Again, where does RNA polymerase know where it needs to begin? It has to start at that promoter region, because that really signals here's the beginning of the gene that we need to copy, and so this is the part that needs to get copied off of. When the RNA is done transcribing that gene, it's going to detach from the DNA once it gets transcribed, and then the RNA does get processed before it leaves the nucleus. There are some segments in the RNA, introns and exons. Introns are segments that don't code for protein, so they have to get cut from the RNA. In other words, they stay in the nucleus, they never get to leave and go to a ribosome, because all we want is the RNA to code for a protein. So if we have these intervening sequences, that's why it has an intron, we need to splice those out of there. The exons are segments that are transcribed that do code for a protein. Okay, so we can say that the exons exit the nucleus. They're the ones that are going to go to a ribosome because they contain the code that's actually going to code for proteins. So a little bit of processing before that messenger RNA can leave the nucleus. There are some other things that happen to it as well, but we focus more on that in AP biology and in genetics, if you would take that. So let's do a little practice here and see if you remember all these different parts. Now, when the DNA separates, remember it's the helicase that unwinds and separates DNA. Okay, I don't really have a picture of it here, so I can't really help you with that. But the two strands that are separated there, this is the coding strand of DNA, and this is the template strand. Okay, so this is the RNA polymerase that's going to go down and copy off of the template strand of the DNA. Where does it know where to start? It has to always find this little promoter region, because it's only going to copy off the segment of DNA that will code for a protein, which is called a gene. And the promoter always signals the beginning of where the gene is. So that's where RNA polymerase knows where to attach. Okay? So this is going to be your messenger RNA that's being copied. This was your original DNA strand here that was being separated. Okay? So make sure you understand the process of transcription. And again, transcription is simply copying of the DNA code onto messenger RNA, because DNA cannot leave the nucleus. This process of transcription occurs in the nucleus. DNA can't leave the nucleus, but proteins are made in the ribosome. So thank goodness we have messenger RNA that can copy that code and carry it to a ribosome where we can make a protein.